Well, I'm delighted to be with you today and appreciate the invitation from the organizers. Novartis is a company of about 100,000 employees. We're located in about 110 countries worldwide, and we're a drug discovery company. And as Bernie Darzinski indicated to you, that's a pretty difficult process. For every 100 projects we start out with, only one gets through at about 15 years, and this costs roughly about $1.5 million. Recently, we've got some good press at Novartis in the last month from Barron's, Fortune magazine, and I forget one other one. We were named the #1 pharma company of the year this year and the pharma company of the decade. Now, that all sounds good if you can actually believe the people who are writing the articles. I'm not sure that I really do. But one thing that we are doing correctly is that we have a real commitment to research, and in this context, a commitment to imaging research. We currently are spending somewhere between $5.5 and $6 billion a year just on the research that we're doing at Novartis. And this really is comprised of a wide array of disease areas, which include oncology, where we're using many of the techniques that you've already seen today to look at size, to look at vascular leakage and so forth. Neuroscience projects, where we're looking from everything from neurodegenerative diseases to other things. We're doing a lot with cardiac, and in particular with cardiac, we're looking at off-target effects for drugs, or things that drugs aren't supposed to do, and looking at diastolic function and hypertrophy in the heart. Also, we're doing a lot in ophthalmology, in treating wet macular degeneration, muscular cell disease, looking particularly at sarcopenia and cachexia, and respiratory disease. We're doing a lot with a lot of things that you've already seen today with the lung, which is very similar, and gastric intestinal disease, where sometimes we link up with those people doing those diseases that have to do with bacteriology, and ATI, or autoimmune transplant, and inflammation, and many of the things that you've heard about, particularly from Dr. Darzinski, in looking at the inflammation of the joints, and basically the compounds and the pathways that can be affected to look at those. And then finally, infectious disease. Well, the way that we approach that, we use a wide range of equipment, and this actually slide is about 3 years out of date, but I throw it on here just the same. So we're using magnetic resonance, which I think through the talks today most of you are familiar with. We also have in vivo whole body optical systems. I have about 6 of those. We also have a PET SPEC CT system that we're using. We're doing a lot of confocal microscopy, particularly with 2-photon capability, and we're doing quite a bit now of life microscopy. In order to do this, we not only have to have the things that we normally think about that are necessary for imaging, but we also have a probe-making laboratory. And in this probe-making lab, we can concoct contrast reagents or small molecule or biological probes, which can actually attach to the targets of the disease. And we can do that for magnetic resonance, we can do that for fluorescence, we can do that for radioactive PET studies. And so if we think about, what we're trying to do in the context of what we're looking at, we're looking to validate the target. In a drug discovery, what the target is talking about is what does the molecule that's going to cure the disease bind to. And we want to know how it binds. We want to know its distribution in the system. We do a lot with disease phenotyping. We're looking about what the drug efficacy is or how effective it is. We're also looking at, oh, that's interesting. Well, I'll just leave it there. Who cares? These slides aren't really very important anyhow. We're looking at pharmacokinetics. In other words, once a drug gets in, can we track it with one of these imaging methods and actually see how it's broken up? And then finally, to look at biomarker development, and hopefully we can translate this into humans. And I'm out of time, and what I'd like to see in the future is the way to image targets, and be happy to talk to any of you, and thank you very much.